Today, I'll be speaking on the quality control of infectious diseases testing in collaboration with my colleague, Joe Vincini. So I'll be presenting the first half, and the second part of this presentation will be a recording from Joe Vincini. So just a quick introduction to the National Serology Reference Laboratory in Australia. We're a scientific, not-for-profit organisation, and we exist to promote the quality of testing for infectious diseases globally. We partner with a broad range of Australian and international partners and really work closely with blood services and diagnostic laboratories and test kit manufacturers. As mentioned, we are a WHO collaborating centre and a WHO pre-qualification evaluation laboratory, and are accredited or certified to a range of international standards. So when you or one of your family members go to a doctor and get a pathology test, you'll get a report similar to this. Each of the different analytes, and here we're showing clinical chemistry analytes, inert chemicals like urea, potassium, sodium. And each of these analytes are measured in units, and some of these units are traceable to SI units. And in a normal human, we have a reference range. And if we test those samples over time, we can see a rise and fall of some of these analytes. And when the analytes are unexpectedly high, we are able to highlight those to the clinician. But when we test a sample in a clinical setting, we're testing a single sample on a single test on a single day. But what if we test that same sample again on the same day, or 10 times on the same day, or after appropriate storage, test that same sample in a year's time on different instruments in the same lab, in different labs using the same assays, or in different assays and different laboratories? Would we get the same results? And are there any differences in the analytes that we're testing? For clinical chemistry analytes, we're looking at inert chemicals, like I said, glucose, potassium, urea, enzymes, like cardiac enzymes, or drugs of abuse or drugs or therapeutic drugs. Where in infectious diseases testing, we're looking for antigens or antibodies, or RNA or DNA associated with that organism. All of the methods that are published for the standardization and control of testing have mainly been developed for Class A analytes, so for inert chemicals. So inert chemicals like glucose have minimal heterogeneity. All of us have exactly the same glucose circulating in our body. And when we measure that analyte, we're actually measuring the amount of that analyte in your system. But in infectious diseases, serology in particular, we're actually measuring the ability of that antibody to bind to an antigen. We're not actually measuring the amount or the number of antibodies. And these are measured using functional biological activities, and these are generally not traceable to a SI unit. So when I started in infectious diseases serology, we used biological assays, so hemagglutination inhibition, complement fixation, where we used a signal, a biological signal like the homolysis or the lysing or the agglutination of a red cell to indicate the presence or absence of antibodies. But over time, we have had the introduction of immunoassays, and which have been converted now into large throughput analyzers. So what's happened in both blood services and in diagnostic laboratories is that the testing for these antibodies have moved from specialized microbiology laboratories into the core laboratories. And these core laboratories are generally under the control of clinical chemistry. And what we find is that the use of clinical chemistry methods for controlling assays are applied to infectious diseases testing. But what I'd like to try and do today is convince you that there are significant differences between clinical chemistry and infectious diseases serology. We had a response to a publication where the respondent said that serological assays are just another assay that follow the same laws of any other. And we feel that this is absolutely not true. So I'll try and explain some of that in this presentation, but you can read more if you're interested in this review paper that we published just a few years ago. This is a list of the differences between clinical chemistry analytes and infectious diseases analytes. When I talk about clinical chemistry, I'm talking about inert chemicals like glucose, potassium, and urea. I'll go through some of these. So first of all, if we take all of the people in a city and test them for glucose, we'll get a standard curve like this one, where the vast majority of people will fall within the normal reference range. But there's two medical decision points, hypoglycemic or hyperglycemic. Whereas if we take all the people in a city and test them for infectious diseases, we're going to get two populations, those that have antibodies and those that don't. And so there's a single decision point, positive or negative, or detected or not detected. So as I mentioned, inert chemicals have minimal heterogeneity. They're the same in all different individuals. We can actually describe them by a chemical formula. We can have, we can trace these, the amount of these antibodies to an international standard using a certified reference method. And these results are often expressed in SI units. Whereas with antibodies, we have variable classes and subclasses. The test could to measure for IgG, IgM, total antibodies, and those antibodies may be free, or they may also be bound to antigens. And each different analyte has, each different organism has different antigenic targets. The individuals have different immune responses, and that immune response changes over time. The assays are designed with different antigens and different antibodies, and those antibodies may be fragmented, polyclonal, or monoclonal. So just to take a view of this image on the right-hand side, you can see this is a Western blot. Each of the strips is a different individual, and you can see the variation or the heterogeneity of the immune response from each of those individuals. But when you test any of those samples from each of those individuals, you'll just get a positive HIV response. But it doesn't mean that this person is, each of the individuals is having the same response. They're not. They're all responding differently to the different glycoproteins and proteins of the HIV virus. And we see this with all of the different viruses, different genotypes of hepatitis C, dengue virus, influenza. And we know that the different individuals can respond differently with their immune response. So some people are immunosuppressed, others are not. When you're infected for the first time with a virus, you get a primary antibody response, which is have very low avidity and affinity antibodies. But these mature over time. So then when you get re-exposed with that virus, your body will respond with a secondary immune response, which has a very effective high affinity response from the memory cells. We know that our immune response to viruses elicit different antibodies to different antigens, and I've just expressed some here that are commonly detected for hepatitis B and for EBV. And we see that the antibodies will rise and fall over the period of the disease. So unlike glucose, where it's homogeneous and stable over a long period of time, antibodies rise and fall. So when we talk about assay design, we know that the antigens that are used in the assays could be from various different sources, or they could be recombinant. The antibodies can be from lots of different species. They can be variable in classes. They can just be fragments, FC fragments of the antibody. And we have different chemical reactions to detect our results. So why is this important? It's important because the traditional methods for establishing quality control assume that the analyte is homogeneous, that the test measures the same analyte over time, and that there's little or no lot-to-lot reagent change. But in infectious diseases, we know that this is not true. So we would argue that the traditional methods for quality control are not applicable for infectious diseases. So we've addressed this in a recent publication for just last year. My colleague now will take over. This is a recording, so he is not present, but he has recorded the rest of this presentation.
Thanks, Wayne. So variation occurs in all forms of testing, but nowhere is it more apparent than in infectious diseases testing. This is because the sources of variation have a major impact on what we see coming out of infectious diseases testing. Reagent lots, instruments and equipment used, how they're maintained and calibrated, the operators and how they handle those instruments and reagent lots. The storage and transport conditions of all the materials that are used and the environmental conditions all have a great impact on the results that we see in infectious diseases testing. And thus one way to understand what this variation means is to use quality control. And this is what we do in routine clinical chemistry testing as well, but they have a different effect in infectious diseases testing. But the principles are fairly similar to start with. So QC samples are the tool that we use, not the end point. So we use those as a method to understand what that variation is, but not giving us necessarily the final answer. And the way we do this is to collect results after each test run. We have some form of software to display those graphically, whether it's Excel or a proprietary third-party software, and have some scientifically validated acceptance rules to understand what those results mean, and then have a documented procedure to handle those unexpected results if they are detected. Using traditional methods with a normally distributed population of data, we would see the example here displayed where we would expect most results to fall within 1 to 2 standard deviations, and anything outside of 2 standard deviations is very unexpected. The traditional methods for setting QC limits rely on mean and standard deviation to set that range. But the question is, what population do you use to establish that range? What data set do you use? And so the data sets set out by traditional methods to be used to establish those acceptance criteria are typically very small, ranging from 15 to 20 data points to establish those acceptance criteria by calculating a mean and standard deviation, and then using some form of statistical model to identify errors outside of those acceptance criteria. When we look at any set of data from infectious diseases testing, in this case HIV antibodies, we typically see results that vary primarily due to lot variation. And if we take the entire data set and calculate mean and standard deviation, we would see that most data would fall within 2 standard deviations as expected. And we would also see some outliers, as we expect 5% of results to be outside that. But if we were to use the traditional method, what we would use is the first 30 or so results to establish mean and standard deviation. And thus, when we calculate mean and standard deviation of the first 20 or 30 results and then apply those to the data set, we can see that the results fall within 2 standard deviations, with a couple of examples right on the borderline of 2 standard deviations. And when we apply those 2 standard deviations we have calculated to the entire data set, we can see that due to lot variation, most of the results in infectious diseases testing would at some stage fall outside of those 2 standard deviations. Thus, scientists find it difficult to use the 15 to 20 results as recommended by guidelines for the traditional approaches to QC, because those 15 to 20 results are not predictive of future results for testing systems that have enormous amounts of variation. That variation, particularly seen with lot variation, is occurring too regularly to be able to be controlled in a manner that is suitable using traditional approaches. The 15 to 20 results do not include sufficient normal variation to provide ranges that are indicative of testing systems that are out of control versus testing systems that are typically in control. And so this creates much confusion for the laboratory scientists. When observing the performance of any data set in testing, the laboratory scientist has a number of choices to make when observing data that are out of control. So what is it that the laboratory does? What actions do they take to rectify any situations where data are out of control? Those actions are varied and depends on the laboratory's experience, but that often includes retesting the QC sample, often many times, test a new vial, open a new box, recalibrate the assay and to retest the QC, refer to a senior scientist for review and often override any results that are out of character, blame the QC sample, call the manufacturer, call in engineers to service instruments, which is very costly and time consuming. When retesting QC samples, retesting usually will not resolve the issue. And if it does, assay performance should be questioned. But more often than not, it does not resolve the issue. And this has an implied cost of reagents, QC sample that's used, the time delays, all costs. And at the end of the day, there's patient reporting and results that is impacted as well. Recalibrating assays may be effective, but often is not as well. Variation is mainly due to lot variation, and so recalibration doesn't often correct this lot variation. It causes significant delays, adds additional costs, but often the kit controls are in range, and so why recalibrate when the kit controls are in range? It is often the case that QC samples are blamed where there is variation observed. QC samples can fail, but rarely are they the cause of variation. QC failure is usually pretty obvious. And in this case, it's good to have a QC manufacturer that is recognized as minimizing QC lot-to-lot variation so that the QC does not become the source of variation itself, that uses or provides peer review software that allows comparison of other reagents lots, and also looks at other assays to better gain understanding of the QC performance, not just in the assay in question, and that the QC provider provides assistance in investigating on your behalf, has access to scientific understanding of infectious diseases, not just of QC in general, and also has access to the reagent manufacturer's scientific affairs to get a better understanding of how the assays work. Here we see an example on the left where the QC is being questioned as to performance, And on its own, this may be a good assumption, but when looking at another assay manufacturer's data, we can see that their data are typically within range here. The blue and the red lines represent different instruments using the same QC, whereas at the same time period, the assay on the left is not performing well, and so it's indicative that the assay is likely to be a major source of variation there rather than the QC. When contacting an assay manufacturer, this option is often only available to large organisations that have good relationships with the assay manufacturers. The investigations take a lot of time, and what are the immediate actions that can be taken? What's the success rate of any investigations that take place? Typically, these are relatively poor in finding root cause, especially if the assay is performing as expected by assay manufacturer expectations. Typically, their kit controls are valid. Can reagent lots be quarantined? Try an alternative reagent lot, and usually that fixes the problem. Is it really the assay that's the problem, or is it the acceptance criteria that are being used to understand variation for your QC? When it's the acceptance criteria that we're using, that is the likely reason why we're observing issues with variation. It's typically because we're re-establishing those acceptance criteria. And what happens if you re-establish acceptance criteria by reagent lot is that we're normalizing the bias we're seeing. We're expanding that range based on observed bias rather than using a meaningful way of calculating what that range should be in the first place. Our approach to QC infectious diseases testing is to use a comprehensive QC program that is specifically designed for the complexities we see with variation in qualitative data that we see in infectious diseases testing. Such a program is QConnect. So QConnect is a method of incorporating QC design with sophisticated software that allows for capture of data and incorporates acceptance limits that are suitably designed for the type of testing we see in infectious diseases. QC samples used in such a program would be ideally human plasma based, optimized for the assays, the test kits that are used. They're manufactured using the same stock material, and that allows for predictable plot variation for the QC so that the QC doesn't become the source of variation and can be used for long periods of time. But the key to all this is to have minimal lot-to-lot variation. The software system is designed to allow for capture of data from multiple laboratories within the one web-based system, and allows for the laboratory to compare their own results using Levy-Jennings charts, but also compare their results with others in a way that allows them to show peer group comparisons of data and able to generate tables and graphs as well. The acceptance criteria established in the QConnect program is referred to as QConnect limits, and these are acceptance ranges for any specific QC assay combination. They're based on historical data and account for the sources of variation that are normally including reagent lot variation. And these have been established to show variance between and within QC lots, and can be used as a trigger for investigations of any form of questionable variation in any testing. Here we can see a number of QC lot numbers that have been tested on a specific assay, roughly 80,000 data points in total, and showing that the mean of results are fairly established, and that allows for good QConnect limits acceptance criteria to be set for an assay across multiple reagent lots and multiple QC lots and multiple instruments, establishing the range effectively for this assay QC combination at 2.1 to 3.6 sigma cutoff range. Thus, when looking at a peer group data chart for the same assay and QC combination, we would see that the established limits for that assay QC combination will be those two red lines, and the majority of data would fall within those lines, and we would consider that to be acceptable normal variation of reagent lots, because historical data predict that reagent lots generally fall between those two lines. NRL is published on the 4 methods that are used in guidelines to establish traditional acceptance criteria in comparison to Q-Connect limits, and have found that Q-Connect limits are far superior in understanding variation with regards to infectious diseases testing, because it was specifically designed for that purpose rather than using guidelines that were not designed for infectious diseases in the first place. Thus, in conclusion, infectious diseases testing is clearly different into testing of inert chemicals such as that found in chemistry. And ID analytes are heterogeneous and vary from assay to assay. Traditional QC methods that are used in clinical chemistry are not fit for purpose for infectious diseases testing. And that is primarily because ID reagents have a lot of lot variation that are not accounted for when using traditional QC methods. And so this leads to unnecessary investigations, waste of time and resources, and QC rejections that may indicate poor QC processes rather than poor assays. The Q-Connect concept designed by NRL is a better approach to QC infectious diseases testing, as it uses historical data to establish acceptance criteria, and that includes the normal variation we see with lot variation. It is supported by EDCDA, a powerful tool to allow analysis of QC data. It is the only scientifically validated QC approach for infectious diseases testing, and is supported by multiple peer-reviewed publications. Essentially, it saves you time, resources, and more accurately identifies unacceptable variation. Both Wayne and I thank you for your attention to our presentation, and we'd be happy to take questions if time permits. Thank you.